Recombinant DNA is formed when a gene from one species' DNA is cut out and inserted into the DNA of another species. The organism that receives the new gene is called a transgenic organism. The transgenic organism will then be able to express that gene. Millions of transgenic bacteria can be grown in a vessel called a bioreactor. Bacteria have two types of DNA, the DNA in their chromosomes and plasmids. Plasmids are naturally swapped by bacteria, so biologists started finding ways to swap plasmids from one bacteria to another. Then scientists discovered two types of enzymes, restriction enzymes that cut DNA at different or specific points, and ligases that join the cut ends of the DNA molecules. So biologists now could insert the gene into a plasmid and transfer the plasmid to bacteria. Insulin is a hormone made by the pancreas to decrease glucose levels in the blood. Before genetic engineering, insulin was only available from animal pancreases and did not have the same structure as human insulin or did not give the same amounts of control on glucose levels. But now, bacteria are grown in large vessels called bioreactors where we can harvest large amounts of human insulin. Some other products of genetic engineering includes enzymes for the food industry, enzymes in washing powder, human growth hormones, blood clotting factors, and proteins that help destroy cancers. A vector is the means of transferring the gene. Along with plasmids, we can also use viruses. A bacteriophage is a virus that attacks a bacterium by attaching to the cell wall of the bacterium and injecting its own DNA into the bacterial cell. So biologists figured if another gene is inserted into the virus's DNA, the viruses will inject it into the bacterium as well. Although most genetic engineering takes place with plasmids, there has been an increase in the usage of bacteriophages as well. Bacteria only has one cell, so gene transfer happens between one cell to another. But plants have billions of cells, and to genetically modify a plant, all cells must receive the gene. So the two steps when it comes to genetically modified plants is 1. Introducing the new gene into plant cells, and 2. Producing a new plant from a few cells. After scientists discovered bacterium, a soil bacterium that inserts its plasmids into cells, scientists had found a vector for the gene transfer. However, this technique would not work for all plants like cereals. So scientists invented a gene gun that fires tiny gold pellets coated with DNA to be transferred. The gene gun can be used to genetically modify cereal plants, tobacco, carrots, apple, and more. Plants have been genetically engineered to have longer shelf lives, to be resistant to weed killers, use genes from Arctic fish to make plants frost resistant, and more. Gene guns have been used to produce golden rice. Golden rice has three additional genes, two from daffodils and one from a bacterium. This allows the rice to make beta carotene, which is converted into vitamin A when eating.